Hello, this is Seher from Easy Peasy, and today we are going to discuss the globular and fibrous proteins part 2. I will put the link of part 1 in the description if you didn't watch that video, okay? Now, in this video, we are going to discuss fibrous proteins in detail. In my last video, we discussed the difference between these two proteins and globular proteins in detail. Okay, so let's start. Fibrous proteins. Fibrous proteins are made by the combination of specific amino acids into regular secondary structure elements. For example, collagen and elastin. So keep in mind, okay, fibrous protein does not have any tertiary structures or quaternary structures. They just have the secondary structures there, okay? Now, if I take the example of a skin, you can see the collagen and elastin is present in it. This is the picture for blood vessel, and this is elastin, this is collagen, and this is the picture of the eye, and in the sclera and cornea, we do have collagen and elastin in it. Now, let's discuss collagen in detail first, and then we will discuss elastin in detail later on. I also made a 5-second quiz for this video and my last video. So please, if you want to take the 5-second quiz in the end, please do watch the first part as well. Okay. Now, collagen. Collagen is the most abundant protein present in our body. It has a structure of rope-like triple helix. So look at the picture. You have an orange alpha helix there, a blue alpha helix there, and a green alpha helix there, and they are coiling around each other and making a braid kind of structure, right? Now, this structure is present in a lot of different parts of the body, but their alignment is also different. How? Just take the example of eye here. If I'm talking about vitreous humor, because it's a jelly-like substance there, the collagen molecule is gonna scatter there and float there, helping the jelly to be stabilized, okay? If this collagen is present in cornea, then it will be in a stacked form to provide stiffness and protection there, okay? If it is present in tendons, then they will have a wave-like structure. You can see over there a different specific pattern for it. So each collagen, according to the type of body it is present in, it can modify itself. Okay, so we have a lot of different type of collagen. We will discuss some of those types here. So I categorize the types of collagen into 3 different things. It's called fibril forming, the network forming, and fibril associated. These are the pictures you can see. Network forming have a mesh. Fibril forming are straight alpha helix structures there, and fibril associated means something is associated with these fibrils. Now, fibril forming have type 1, type 2, and type 3. Network forming contains type 4 and type 8. Fibril associated collagen will have type 9 and type 12. Now, type 1 is present in tendon and cornea. Type 2 is present in cartilaginous structures, and type 3 is present in blood vessels. The network forming collagen is present in base membranes, and fibril associated collagens or proteins are present. It binds these fibrils to one another and other components in the extracellular matrix as well. Now let's discuss the structure of collagen in detail. We know it consists of 3 polypeptide chains coiling upon each other. Now we will take only one polypeptide from it and see what are different amino acids present in this polypeptide chain. Okay? So the most common amino acid, like every 3rd amino acid that is present there, is glycine. So glycine is the most abundant amino acid present in this polypeptide chain. Okay. The second most abundant polypeptide amino acid is proline here, and the 3rd one is hydroxyproline, or it can be hydroxylysine. it's 70% chance the hydroxyproline and hydroxylysine is present there, but 30% chances there can be any other amino acid there. But these are the most common one, okay? Now, in my last video's name as 4 level of proteins, I guess so, I did mention that proline is basically a helix breaker, or it is not compatible with alpha helix. Remember, because of its side chain, the amino group is not available, so it's not compatible with the alpha helix. And now in this slide, I'm saying that proline is the every second amino acid there, right? So I am conflicting my own statement. No, I'm not conflicting. When I was making this video, this question comes in my mind, and I'm going to answer right away, so your mind should not get confused about these things, okay? The alpha helix that's going to generate by the proline molecule over there is not straight. It is in this kind of structure, and we use the terminology kink for these kind of structures. Okay, so these kinks are basically produced by the proline in the alpha helix structure. So I'm right, proline is alpha helix breaker. That's why the alpha helix is not straight, it is having kinks in it. Okay, according to me, it looks like somebody's having a back pain. Fine. Okay, now which bond is stabilizing this alpha helix? Now let's see. Glycine belongs to nonpolar side chain group, right? And as far as I remember, proline also belongs to nonpolar side chain. As far as lysine is concerned, it is present in the basic side chain, but this is not the lysine only. It has hydroxylysine, right? So the most common bond that is stabilizing this alpha helix is hydrogen bond. Make sense, right? Okay, now we have 3 different polypeptide chains having kinks in it, and they are coiling upon each other just like you're making a braid of your hair. Okay, bingo. Let's go for the next slide. Now let's see how collagen is synthesizing in our body. So the cell that usually plays a role for synthesizing collagen is called fibroblast. Now I'm not gonna talk about how the nucleic acid is transcripting messenger RNA and how messenger RNA is translating into proteins. I'm just gonna discuss the post-translational modifications that's gonna occur after the protein is formed. Okay, so we are not going in the nucleic acid part, we are just going to focus on the protein part there. So let's start. Here we have nucleus and it creates messenger RNA. Messenger RNA then run towards the apartment called rough endoplasmic reticulum, where it's gonna translate into protein. Now, the protein that is going to be translated in rough endoplasmic reticulum is called a pre-pro-collagen. Okay, now we know which amino acid is present there. 90% glycine, 80% proline, and 70% it can be lysine or it can be proline, right? I didn't use the word hydroxy right now. Why? Because that's the post-translation modification. When the protein is formed just in the endoplasmic reticulum, hydroxy group will not be attached to it. It's going to attach now, okay? Now to attach hydroxy to this lysine group here, we need enzyme, and that enzyme is lysyl hydroxylase. If the third amino acid will be proline and we need to attach a hydroxy group with it, then prolyl hydroxylase will be there. Okay, in this situation I put lysine, so lysyl hydroxylase is going to attack the lysine here. Okay, now this enzyme need a backup or need a help, and the help is provided with vitamin C there. And with the help of vitamin C, lysine is going to transform into hydroxylysine. Now this vitamin C is really important, okay? If you have less amount or less quantity of vitamin C in your body, and it's not available to transform lysine into hydroxylysine there, the person will have a disease named scurvy. And these are the different symptoms there: bleeding in the gums, rashes around. I'm just going to remove the pic. I really don't like these kind of symptoms, okay? So vitamin C is really important, and vitamin C attaches hydroxy group to lysine and prevents scurvy in our body. Bingo, right? Now the whole process is called hydroxylation. Okay? Now, we have the structure: glycine, then proline, then hydroxylysine. Now, the next post-translation modification that's going to occur here is the attachment of glucose or galactose with it. And this is the symbol I just created for it. So this is going to attach with that lysine group, and this process is called glycosylation. Now this process is also very important. At this point, the collagen, the pre-pro collagen, is called pro collagen only. Okay, at this point, the 3 polypeptide chains we know now is going to coil upon each other due to the help of this glycosylation. Okay, if there is some kind of defect present here in glycosylation process, then we have the disease called osteogenesis imperfecta, and the symptoms of this disease is like this: blue color in the eyes, your backbone will not be straight, it will be curvy there. Okay, again, I don't like symptoms, I'm going to remove it. So these are the 2 post-translation modifications occur when collagen is going to transform. The process is not ended right now, but I need to fantasize it so I should not forget it, right? According to me, what happened is, I have a person called Gopal. Why I use the name Gopal here? Just pay attention. G for glycine, P for proline, and L for lysine. So I just add them up and made a name, Gopal, for me, okay? Now this Gopal guy went to a restaurant and he take the orange juice there. Why orange juice? Because it has water and vitamin C in it. So this process is basically hydroxylation for me. Okay, and then he eat a donut there, so he is actually having glycosylation. Now Gopal, after eating and drinking these 2 things, does not remain same, right? So this pre-pro collagen Gopal will transform into fat Gopal there. Easy, right? So just imagine a person having orange juice and a donut, and you will remember the post-translation modification of collagen there. Bingo, right? Okay, now we know when the glycosylation process is occurring there, the 3 polypeptide chain is coiling upon each other. But look at the picture. It is congested at the C-terminal. Why? Because they have disulfide bonds at the C-terminal. Now the question is, I said the polypeptide chain should have glycine, proline, and lysine. None of these amino acids have sulfur in it. So how these disulfide bonds are present at the C-terminal, right? The question does come in your mind. The answer is, remember I said 70% chance? It means 30% chances you might have methionine or cysteine there, or any other amino acid there, right? So at the specific portion of the C-terminal, they do have methionine and cysteine group there. Those amino acids do have sulfur group in it, and those sulfur groups are making disulfide bonds there. Make sense now? Okay. Now the disulfide bonds are made, and we are in this section here. Now what will happen is the cleavage will occur. So the cleavage will occur from the non-coiled pore from the C-terminal and non-coiled pore from the N-terminal as well. So once the cleavage is occurring, this triple helix is called tropocollagen now. So I use 3 terminologies, okay? Pre-procollagen, the first protein that is originated from the rough endoplasmic reticulum without post-translation modification. Procollagen, when post-translation modifications are done, but trimming is left. Once the trimming is done, then this protein is called tropocollagen, okay? Now, tropocollagen will make cross-linkings with each other, and the enzyme that's going to help it is copper-containing lysyl oxidase. So it means the lysine amino acid that is present in the polypeptide chain is basically helping in making cross-linkings there. Make sense? Right. Okay. Now let's go for the elastin. Elastin is the second most important protein that we are going to discuss in this video. Elastin is a connective tissue protein with the rubber-like properties. They can be stretched to several times their normal length, but recoil to their original shape when the stretch force is relaxed. So just like look at the rubber band there, you can stretch it, you can relax it, so it's coming back into its original shape. Bingo. Now, elastic fibers. Just remember, elastic protein is present in those parts of the body which can move, like my jaw while I'm speaking, my hands while I'm using or picking something up, my foot when I'm walking, my lungs because they are constantly contracting and relaxing to pump the air, right? My heart because it's continuously pumping the blood in my body. So every part of the body that can move, my eyes, they are blinking, right? They have elastic fibers in it. So elastic fibers basically consist of elastic molecules and microfibrils in it. Again, they will have cross-linking in it. Again, the enzyme here is lysyl oxidase. So just keep in your mind, lysine is the amino acid that is making the cross-linking there. This is the area you can see cross-linking when the situation is relaxed, and this is the situation when the cross-linking is there and the situation is stretched. Okay? Elastin is also rich in proline and lysine, but are deficient of hydroxyproline and hydroxylysine. So the post-translation modification we discuss in collagen are not present in elastic fibers that much. Now let's see the synthesis of elastin here. Now this green portion is basically a cell. This black portion is basically the extracellular matrix you have, and this other, again, the tiny black portion here is basically showing you that these elastin proteins are coming from a tropoelastin molecule. Now these tropoelastin molecules are going out of the cell, right? And they are taking help from these elastin binding proteins. So these purple things are attached to this protein, helping them out to go out of the cell. And once the tropoelastin is out, these proteins are coming back in the cell, so they can be recycled, you can say. Bingo. Now these are tropoelastin, and they have more fibrillar proteins here. These fibrillar red lines are gonna stack with it. Now, in the last slide, I said microfibril and elastin molecules are gonna combine with each other. So here we have microfibrils. You can see in the form of these lines with the dots there. And these are the molecules of elastin. So these molecules are gonna stack here within microfibrils and making the elastin protein for us. Now what is left? Lysyl oxidase, the enzyme that is gonna make the cross-linking in between, right? That is left. So enzyme is coming, and these cross-linking you can see is here. And now this perfect molecule is shifted into elastic fiber. Bingo! Now, the thing that is important in this step, the fibrillin. If these fibrillin proteins are not attached properly, or there is some kind of problem in it, the disease we will face is called marfan syndrome. And these are the symptoms of marfan syndrome. The fingers are not in a normal size, they're more elongated. I'm going to remove the picture. Again, if the cross-linking is not sufficient in this elastin protein, it can cause aging. So with the passage of time, when you're young, the cross-linking is perfect, but with the passage of time, these cross-linking is going to reduce, and once they reduce, the relaxed portion remains relaxed and the contracted portion remains contracted, and you can see wrinkles on your face. So that's how the aging comes in our face. Okay, now let's get ready for 5-second quiz. Okay, my first question: what is this yellow circle in the picture? 5, 4, 3, 2, 1. This is heme protein. Next question: what is the blue circle in the center of the heme group? 5, 4, 3, 2, 1. This is ferrous atom or iron atom, you can say. Next question: heme is present in globular proteins or fibrous proteins? 5, 4, 3, 2, 1. It is present in globular proteins. Remember the myoglobin and hemoglobin we discussed in the last video? Okay, next. What is the role of 2,3-biphosphoglycerate in heme-heme interactions? This is your clue. 5, 4, 3, 2, 1. It's going to detach the oxygen molecule from the heme group. Remember, this is a wedge. So wedge is going to disrupt the marriage. Okay, next question. Scurvy occurs due to the deficiency of what? 5, 4, 3, 2, 1. And the answer is vitamin C. Next question. What is the function of vitamin C in the synthesis of collagen? 5, 4, 3, 2, 1. It helps in hydroxylation of lysine or proline. Remember? Next question. What is the most abundant amino acid present in collagen? 5, 4, 3, 2, 1. Glycine. Next. Which amino acid produces kink in the polypeptide chain? 5, 4, 3, 2, 1. Proline. Okay, next. Which amino acid provides cross-linking in collagen and elastin? 5, 4, 3, 2, 1. Lysine. Thank you very much. If you like my video, please give me comments that how you find it and subscribe my channel. Thank you. Bye bye.